he's been doing.
Thank you so much, Brent. It's really a pleasure to be here. I'd love to tell you today about the work that we're doing, translating genetic findings into mechanistic insights that can enable new therapeutics. And in particular, how do we not only dissect, but also manipulate the circuitry of human disease at single cell resolution? So it all starts with genetics. Why genetics? Because genetics gives us causality, it gives us mechanism, it enables us to find completely new pathways and new target genes that were previously unsuspecting. It gives us the ability to design completely new therapeutic avenues and to enable ultimately precision medicine and personalized medicine. It starts with both common variants and rare variants. And in particular, the workhorse of human genetics for the last at least decade and a half has been genome-wide association studies that are looking across the entire genome at roughly 6 million independent SNPs in the genome and their association on the y-axis with various traits using a chi-square statistical test. So this is a typical GWAS Manhattan plot where you can see some of the skyscrapers reigning supreme above all others, and then a huge sea of still tall buildings in the New York skyline. Here I'm showing you the GWAS plot, the Manhattan plot for obesity, for body mass index. And specifically, you can see that this FTO locus, which was promptly renamed fat and obesity associated, is in fact stronger than all of the other loci, and it was both the first and still remains the strongest genetic association with obesity. This is a glimmer of hope. It basically says there's something going on in that genetic region that can give us new insights, new therapeutic avenues against what is a plague for our society across the world. The challenge, however, is that when you look at the genetic variants associated with obesity in this region, you see that out of 89 common variants that are co-inherited, all of them sit in the first intron of the FTO gene and part of the second intron of the FTO gene. This tiny little dot here and there are the exons, and none of the genetic variants that are associated with obesity is in fact perturbing the FTO protein directly. And this is not just some random weird exception. This is true for 93% of disease associations from genome-wide association studies. They're all sitting in non-coding regions. What does that mean? That means that we don't actually know that the target gene is this fat and obesity associated gene. We don't actually know that the causal variant is the one that perturbs the gene, since none of them perturbs the gene directly. We don't even know the cell type of action, because to figure out where these regions are active, we need to systematically study their circuitry to study when these switches are active and what cell types they act in and which cell types they exert their effect in. And we don't know the pathways and we don't know the mechanism. And that's the challenge that we have ahead of us. We have this amazing opportunity of 120,000 genetic loci that are associated with complex traits, but this enormous challenge of interpretation. And interpretation means not just understanding the target gene, but understanding the complete circuitry. What cell type it acts in, what target gene, what condition, what SNP, what upstream regulator, what pathway, and so on and so forth. So that's what my research focuses on systematically. We start with disease genetics across both common and rare variants, and then we systematically, in our experimental lab, profile a variation in both the transcriptome and the epigenome of thousands of samples across both healthy individuals from different disease, from different genotypic backgrounds, with and without the risk allele for ApoE4 in this case, for Alzheimer's, and so on and so forth. But also in the context of disease, because not every variation will be genetically driven. Some of that will be driven from the environment, from behavior, from socioeconomic status, and all kinds of other settings. So we basically complement the genomic data with DNA accessibility, histone modifications, DNA methylation, which together form epigenomic signatures for understanding the circuitry of these regions outside the protein coding genes. And of course, the transcription of both protein coding and non-coding genes to understand how this is changing in response to genetics and in response to disease conditions. We then computationally integrate all these data sets across both healthy and diseased individuals to predict candidate driver genes, regions, and cell types that mediate the effect of these genetic variants on the disease phenotype. And of course, to really prove causality, in addition to the causal inference methods that we develop, we then perturb these predictions using cell cultures and using mouse models. And of course, disseminate the results and start the cycle all over again. What do I mean by circuitry? By circuitry, I mean that instead of just saying there's something going on in this region of genetic association, we can say extremely precisely, here are the SNPs. These are the single nucleotide polymorphisms. These are the genetic variants that underlie this association, number one. Number two, these are the enhancer regions within which these SNPs are sitting, suggesting that they might be perturbing these control elements. So enhancers are distal regulatory regions that can act sometimes at 1 million nucleotides away, and promoters, of course, which are the proximal gene regulatory regions. Then we would like to ask, where are these enhancers active across different cell types? And in this particular case, there's a cholesterol association with a liver activity across multiple of these enhancers. Then we would like to know who are the upstream regulators that are responsible for the circuitry of these regions, from the regulator to the enhancer in the specific cell type, and then ultimately, what are the target genes downstream of these associations? So we basically develop methods that do that systematically across the entire human genome, across thousands and tens of thousands of SNPs, to start understanding that circuitry. And then when we understand the circuitry, we can start manipulating the circuitry. So in the case of the FTO locus that I showed you on the first slide, we use these approaches to show that out of the 89 common variants in that region of association, there's a single nucleotide polymorphism, a SNP here, that perturbs this AT-rich motif by changing a T into a C, specifically variant RS142108. That variant is, that motif is normally bound by this AT-rich interacting domain repressor, ARID5B. It binds a 12 kb super enhancer in the FTO region of association that I showed you earlier. And normally that repressor shuts down the expression of two downstream genes, IRX3 and IRX5. These genes are sitting not on the spotlight where people love to look when they're searching for their keys, but instead 1.2 million nucleotides away for IRX5 and 600,000 nucleotides away for IRX3, several genes over. So that basically means that to really understand the target, we need to understand the circuitry. And then what do these genes do? Their function was previously unknown and unrelated to metabolism. But what we showed in our paper is that they are in fact master controllers of UCP1, uncoupling protein 1, and many other proteins, which are leading to different pathways that control thermogenesis, heat generation, a process through which, by depolarization of the mitochondrial membrane, our adipocytes can choose to burn the excess calories in our diet instead of storing them as these high energy molecules that are lipids and then becoming these really big, fat, juicy, white fat cells, which ultimately lead to obesity. So what this variant does is that it acts in the first 3 days of differentiation of adipocytes to switch between a thermogenic and a lipogenic program. The thermogenic program burns the calories and keeps individuals lean. The lipogenic program stores these calories and makes individuals obese. I am a double homozygous carrier of the risk allele. So thank you, mom and dad. None of them is homozygous, only I am. So gee, thanks. And that basically means that if I have excess calories in my diet, I will store them. It sounds like a pretty bad thing to do today because we're fighting with obesity. But you know, a few generations ago, there was no association between FTO and obesity. And in fact, a few generations before that, it was extremely beneficial to have the fat storing allele. And this allele rose in frequency from 2% in Africa to 44% in Southeast Asia and 42% in Europe, suggesting a selective pressure for storing calories. Why am I telling you this? Because I don't want the stigma of, oh, it's disease associated. It's phenotype associated, and phenotype is context dependent. Sickle cell anemia is the bad thing in the US, but the converse is being protected against malaria in Africa. Similarly, in periods of starvation, thermogenesis is a pretty bad idea. Lipogenesis is a great idea. If you're going to McDonald's all the time and just sit at the office all day having Zoom meetings, then thermogenesis is a great idea and lipogenesis is a terrible idea. So once we uncover the circuitry, we are excited because that means we can manipulate the circuitry to reverse the disease phenotypes. And this is what we did. I like to say that we wrote a paper about a single nucleotide in the human genome. And that nucleotide is this one. We were able to show that we can manipulate that one nucleotide to switch back and forth between the lean and obese phenotypes. But we can also manipulate the downstream target genes directly and have a much stronger effect than what nature will tolerate as basic genetic variation in the human population, which is generally a weak effect. So the reason why I'm pointing this out is because many people are saying, oh, great, let's go edit genomes now that we know what the causal variants are. No, editing genomes is really not what we need to do. What we need to do is figure out the minute changes that evolution tolerates within the human population, and then understand the circuitry and carry out much bigger changes at the downstream target effectors or even the downstream pathways, no matter how we target them, because we know that these pathways now have a causal effect on the phenotype. So in our paper, we basically showed that increasing or decreasing either of the two target genes can switch back and forth between lean and obese individuals. Increasing or decreasing the upstream regulator can also serve as a switch. And even the CRISPR-Cas9 genome editing of a C to T or a T to C can switch back and forth between lean and obese phenotypes. In particular, if we take primary adipocytes from homozygous risk individuals that carry the CC allele, who like me are unable to thermogenize both in the basal conditions and in the stimulated conditions, and we turn on this binding site for ARID5B by switching this C into a T that this AT-rich interacting domain loves, we see restoration of binding, restoration of repression for IRX3 and RH5, and most importantly, restoration of the cellular signature of thermogenesis, namely reactive oxygen consumption rate as a percent of basal, which turns back on both in the control and in the stimulated condition. So with a single nucleotide change out of 3 billion nucleotides of the human genome, we're able to restore this pathway in primary adipocytes from obese individuals. In mice, we're able to show that the IRX3 gene, which is shutting down thermogenesis, when expressed from an adipose-driven dominant negative construct, is shutting down IRX3 and then turning on thermogenesis. These mice start out leaner to start with, and then when put in a high-fat diet, the normal mice that are unedited gain weight with a high-fat diet. These dominant negative IRX3 mice are unable to gain weight. If you observe their fat stores, they're simply depleted, they're gone. They are unable to store energy as fat. They're only burning that energy. They don't change their appetite, they don't change their diet, they don't change their exercise, they don't change their activity. They're simply burning calories when they're awake and when they're asleep. So this is what we would love to do for every locus of the human genome. Across 120,000 disease loci, we want to systematically understand the circuitry and systematically manipulate the circuitry. How do we do that? We start with reference epigenomes to understand the disease-relevant tissues, nucleotides, upstream regulators, and mechanisms through which the non-coding genome functions. We then look at variation in this epigenome across different genetic backgrounds. We look at genetic, epigenomic, and transcriptional variation, both in healthy and in diseased individuals. We then look at the single cell level at this epigenomic and transcriptional variation. We combine it with genetic variation using deconvolution and using the single cell datasets directly in the context of both genome-wide association studies and expression or methylation quantitative trait locus studies. We then integrate these phenotypes beyond just the disease, non-disease, to include many intermediate phenotypes through medical records and through additional measurements in these individuals. and we go beyond individual tissues to multi-tissues and understand how these genetic variant effects are converging across multiple pathways to obesity, cancer, Alzheimer's, and aging, and how we can use biomarkers even from peripheral blood to understand these multi-tissue effects. And then we develop extremely high throughput methods for dissecting that circuitry, for carrying out 7 million experiments, 7 million measurements in a single assay, and hundreds of perturbations in a single assay. So I will walk you through these different types of activities, acknowledging each of the papers in the bottom right of the slides. So let's start with the reference epigenome. This is something that Brent and I have worked very closely on in the context of the ENCODE genome project, the Encyclopedia of DNA Elements, and also in the Roadmap Epigenomics projects with many colleagues across the US and actually the world. We basically, in these collaborations, profiled through multiple experimental labs working closely together, many tissues in the adult and in embryonic stages of development, many brain regions, embryonic stem cells, induced pluripotent cells, and in vitro differentiated cells, as well as primary cells from these individuals. We've then profiled many epigenomic assays, including, and these are going to be color-coded, promoter-associated marks, enhancer-associated marks, transcription-associated marks, polycomb-associated marks. This is polycomb repression, this is heterochromatic repression, this is activation in both enhancers and promoters, DNA accessibility, which can be both an indication of binding, either of activators or of repressors, as well as DNA methylation, which is typically a signature of a repression in gene regulatory regions. And of course, the quote unquote output of RNA using multiple methodologies. We've then developed computational methodologies for integrating all of these different signatures into a combined annotation for every tissue and cell type profiled. In 2015, we basically published this epigenome roadmap project across 127 cell and tissue types that gave us the most global view of the dynamic epigenome. Looking at promoters, you could see that they're extremely stable even when the gene is not expressed. Looking at enhancers, you could see that they're much more dynamically changing. Looking at PAX5, for example, we could see that it is repressed in every cell type in gray, except for a handful of immune cell types where it appears to escape repression and turn on. And most importantly, we can now look at gene regulatory regions and then say, where are they active? What could be potentially the tissue of action of genetic variants that are sitting here? And you can see here, these genes, this gene regulatory region, even though there's no gene there, is in fact turning on very specifically in these cell types. And even perhaps more importantly, we can use the correlation between non-coding region activity and nearby gene expression to predict that maybe the target of this region is not just any of these nearby genes, but is instead this PAX5 gene that appears to be turning on exactly in the cell types and tissues where this regulatory region turns on. Armed with this arsenal, we can start systematically looking for enrichment between the genetic variants associated with multiple different traits, looking at both regions of association and individual SNPs that are lying within this region, not just across one trait, but across many traits. Why? Because we can use these different traits as controls for each other. We can basically say, what are the epigenomic enrichments that we're finding for height, and how do they differ from those of type 1 diabetes, blood pressure, cholesterol, and so on and so forth, to infer the tissues where different genetic variants might be acting for each trait. How? By basically saying that across all of the genome, genetic variants associated with height appear to be preferentially overlapping enhancers that are active in stem cells. Genetic variants associated with type 1 diabetes preferentially active in regions in immune cells, and so forth. So this is actual data. You can see here that genetic variants associated with height are overlapping epigenomic annotations specifically active in embryonic stem cells, immune traits, T cells and B cells. Blood pressure variants are overlapping specifically in the left ventricle, in enhancers active in the left ventricle of the heart. If you look at fasting glucose-related traits, which are associated with type 2 diabetes, you see very specific enrichment in the pancreatic islet, where insulin is produced, making complete sense with the biology. If you look at cholesterol, it doesn't matter how you measure it between HDL, LDL, and many other lipid traits, you see that the liver appears to be the place where these enhancers are overlapping these regions. If you look at inflammatory bowel disease, inflammatory, you see a lot of immune cells, and then bowel disease, you can see these digestive tissues very specifically enriched. Both signatures, both tissues appear to be contributing. If you look at Alzheimer's, you would have expected, or at least I would have expected, the brain to be the major tissue driver, where the enhancers would be overlapping genetic variants associated with Alzheimer's disease. But instead, we see no enrichment in the brain. Instead, we see a very specific enrichment for CD14 plus cells. These are the signatures of both circulating blood monocytes and tissue-resident macrophages, which are immune cells in diverse tissues of the body, including the microglia inside our brains, which are a very small minority of cells. They're only 5 to 10% of cells, when the majority in whole brain samples are in fact coming from times of COVID, when the majority of cells are in fact excitatory and inhibitory neurons, oligodendrocytes, astrocytes. Only 5 to 10% of cells are in fact microglia. But that suggests that that's why we don't see an enrichment for the whole tissue, because that signal is hidden in a very small minority of cells. We can test that hypothesis by specifically cell sorting neurons, microglia, and other glia, the majority of which are oligodendrocytes in this assay, and then carrying out the chromatin immunoprecipitation experiment for this enhancer-associated mark, and finding that indeed the genetic variants for Alzheimer's are specifically enriching in microglial enhancers according to this signature. That led us to publish a paper back in 2015, along with a roadmap epigenomics paper that also validated this prediction in mice by showing, in collaboration with Liquid Science Lab, that the CKP25 model of Alzheimer's disease, which develops both early and late symptoms consistent with Alzheimer's progression in human, but in a very short time span, shows immune changes early in the disease progression, but no neuronal changes until later in the disease progression. Again, suggesting that inflammation and immune dysregulation are probably the causal component of Alzheimer's, which only then leads to neuronal dysfunction. So that was in 2015. We basically published the 127 epigenomes as part of a roadmap epigenomics paper. In 2021, just a few weeks ago, I can't believe we're already in 2021, we basically published a new paper expanding this to 834 tissues and cell types, which we call EpiMap, which now cuts across ENCODE, Roadmap, and GGR and other consortia. That has allowed us to expand this matrix from 54 GWAS traits to 534 enriched traits, and to expand to 30,000 single nucleotide polymorphisms that are sitting specifically within enriched enhancers and are associated with diverse diseases and complex traits. This is a quarter of every single genetic region that has ever been associated with GWAS. And of course, we expect many, many of them to be acting post-transcriptionally and in other non-enhancer associated ways. And also in tissues that are perhaps still not captured by this much larger epigenomic map. What we are finding is that indeed Alzheimer's genetic variants are still localizing predominantly in immune cells, along with many other immune traits, while neuroticism, schizophrenia, depression, as well as intelligence, cognitive performance, highest math class taken, are localizing instead in the brain samples, suggesting that they're acting predominantly in neurons that are the majority of the cell types making up the brain. Filtration traits enrich in kidney, cholesterol-associated traits enrich in liver, heart association and heart repolarization, enriched in heart enhancers, and so on and so forth. But in addition to these clusters, which are very densely populated, you also see many traits in the middle, which are what we call polyfactorial, which are acting in multiple tissues simultaneously. The most polyfactorial is coronary artery disease, where you can see enrichments in liver, coronary artery, adipose, thyroid, and many other tissues. This prompted us to now say, well, can we take these complex traits and start dissecting them out, partitioning them into their subparts? The subset of SNPs that overlap liver enhancers and are associated with coronary artery disease are in fact enriching in very different biological functions than the subset of CAD variants that instead overlap coronary artery enhancers, and so on and so forth. That allows us to now start partitioning the complexity of these polyfactorial traits into their parts, both for gene ontology enrichment and for comorbidity and co-association with different types of related traits. And we can do this not just at the global level, but also in individual loci. We can now use this annotation to go down the list of GWAS loci, as I showed you earlier, for 30,000 of these loci, and start looking at how far is that enhancer from the SNP in that region that is marked as the lead SNP in the association. And you can see here some examples of, number one here, PCSK9, which is one of the beautiful stories of success of first a genetic association and now a drug in the market that specifically targets PCSK9. We find PCSK9 as the target of a liver-specific enhancer, which you can see here enriches in liver and liver. And this appears to be a very specific function. And you can also see these visualizations which we have created for 30,000 SNPs in the genome, where you can see here the top SNP is in fact linked to the promoter of PCSK9 and predicted to act specifically in liver. Here's another example. In chromosome 4, this eDNRA genetic locus overlaps coronary artery and heart atrium, and indeed is linked to this eDNRA gene, which is active in the heart with heart-specific links. Here's another example, which at the surface appears to be perhaps a little more pleiotropic. It appears to be active in multiple tissues. And indeed, when we zoom in, we see that it is linked to both a liver in blue and a heart in red gene, with tissue-specific links in each case, suggesting perhaps multiple target genes and multiple target tissues for this region of association shown here. We've made all this data available in a website called EpiMap. If you just Google EpiMap, you'll probably find it. Here's the URL. And you can see these visualization links for 30,000 loci, where you can download any GWAS and then click on the different pictures that are pre-generated, and also navigate in an interactive fashion with the gene regulatory circuitry of the upstream regulators, the enhancers, the tissues in which they're active, and all of the gene ontology enrichments across tens of thousands of these pictures. But in addition to the epigenomic information, we can use comparative genomic information for annotating these genetic variants. We can use these evolutionary signatures of protein coding variation, RNA structures, microRNAs, regulatory motifs that each evolve in very different ways to distinguish where are the protein coding genes, what regions are non-coding, where are the RNAs, where are the motifs, where are the microRNAs, and so on and so forth. And we can use that to now start going into individual binding sites that are perturbed by regulatory motifs and that are predicted to be drivers through this, enabling us to do this fine mapping. But of course, in the time of COVID, we've also used these comparative genomics methods to understand the genome of the enemy that is, keeping us all indoors and unable to have drinks after this keynote address. And this is the entire genome of SARS-CoV-2, the virus responsible for the COVID-19 pandemic. It's 29,000 nucleotides. So you would think that compared to the 3.2 billion nucleotides of the human genome, which is 100,000 times larger, that all of the protein coding annotations of this genome would be resolved. However, there were indeed multiple hypothetical ORFs. And what we showed through comparative genomics of 44 related species in a paper that just appeared last week in Nature Communications is that several of these genes are in fact not real genes. So there was a prediction that there would be multiple overlapping ORFs that we showed are simply not real. So for example, ORF9C, we don't see any signal for protein coding selection. ORF10, which is not overlapping anything else and was predicted to be protein coding, look at how large, how strong the conservation signal is in fact completely non-coding, not a single signature of protein coding selection. By contrast, there's a new ORF here, ORF3C, which is overlapping in a different reading frame, ORF3A, which we were able to predict as a new protein coding gene under dual selection, both in the ORF-A reading frame and in an alternate ORF reading frame for ORF-C. We're able to use this comparative genomics also to look at how fast and how slow different genes are mutating in the current pandemic and across the massive evolutionary timescale that our 44 sarbecovirus genomes are spanning. And what we found is that, for example, the nucleocapsid protein, which is responsible for wrapping together the RNA genome and packaging it up inside the viral particles for transmission, is in fact showing many more SNPs than expected. And many of these very rapidly evolving regions of the nucleocapsid protein are in fact epitopes, suggesting that the mutations might be evading our immune response against the virus. By contrast, S1, which was super rapidly evolving across the sarbecovirus gene tree, is in fact evolving very slowly in the current pandemic. Again, we have multiple theories and explanations for what could be driving this. But other than these exceptions, you see a general agreement between how fast something has been going across different species or different strains and within the current isolates. And lastly, we can use these comparative genomics to start interpreting variants, not human variants, but virus variants, and specifically the D614 gene mutation that was increased early in the pandemic in multiple independent cities is in fact disrupting an amino acid that has never been changed across what I like to call a mammalian equivalent of 60 million years of evolution in a region of 11 perfectly conserved amino acids where only synonymous substitutions are happening. That suggests that it might be a new human host adaptation, or perhaps that early in the pandemic, the mutations that are tolerated are the ones that will increase transmission in a virgin immune system when no one has immunity. And our prediction is in fact that this mutation will actually start decreasing in frequency as we start developing global immunity against it. So everything I've told you so far has been about reference epigenomes to understand the basic circuitry, the basic building blocks of the genome. Where are the control regions? Where are the motifs? Where are the targets? What control regions are connecting to where in order to interpret genetic variation? But we can actually go start looking at the variation in this epigenome across individuals, because everyone in this Zoom channel is in fact having a different epigenome in every one of their tissues than every other person in this room. So how do we do that? We basically want to bridge the gap, which is enormous, between a single nucleotide variant and all the way to a disease phenotype. And there are thousands of genetic variants contributing to disease, each of which has a tiny little effect. However, by bridging this very long path to intermediate phenotypes in specific tissues, specific regulatory regions, specific genes, specific intermediate phenotypes, we can start understanding the path of causality and understanding how the environment is modulating these intermediate phenotypes as well as the disease, and how many of those are in fact downstream consequences of the disease. So the challenge, of course, is that these beautiful unidirectional arrows that we have coming out of genetics are no longer unidirectional. They're bidirectional in the intermediate phenotypes because it's correlation, it's no longer causation. So that requires developing new computational techniques to infer causality. How do we do that? By combining genetic variation, epigenomic and transcriptional variation, and phenotypic variation to start first learning how to predict the epigenome from the genome of a person, and then how to together you look at the correlation with phenotype and specifically the causal component of that variation. So looking at this systematically across 723 individuals from the Religious Order Study and Memory and Aging project, driven by David Bennett at Rush University and in collaboration with Phil de Jager, previously at HMS, now in Columbia, we basically looked at how can the genome allow us to predict the epigenome. And what we found is 50,000 genetic loci, where if I know the genetics of a person at conception, I can predict the methylation of that person at 93 years of age. And this correlation allows us to now start predicting the epigenome systematically. So expanding beyond genome-wide association studies that are looking at this very long path, we can start predicting which genetic variants modulate either the methylation or the transcriptome of these individuals in specific tissues, and then carry out not a genome-wide association study, but a methylome-wide association study, which again would give us a bidirectional correlation between disease and methylation, because some of the methylation changes might be downstream of disease, in contrast to genetics, which is always upstream, especially for inherited variants. But instead of doing this for observed methylation, we can do this for imputed methylation based on this G2M function that we can learn here. That means that we can specifically look at correlations between the genetic component of the disease rather than of the methylation change rather than the full methylation. So doing this systematically, we can now start predicting new regions of association by combining multiple nearby SNPs in the prediction in regions such as chromosome 6 that have no genome-wide significant variant. 10 to the minus 8, 5 times 10 to the minus 8 would be up here above the top of the slide. That allows us to do this not just in methylation, but also across multiple intermediate phenotypes, methylation, transcription directly, and ultimately disease, and start predicting hundreds of new loci. So within genome-wide significant loci, shown in purple, we can predict what are the target genes, what is the tissue of action, and what is the directionality of effect using these approaches. But we can also look into new loci that are not previously genome-wide significant, shown here in gray, and predict additional regions that are now predicted to be causal in the disease. But everything I've told you so far has been at the global tissue level, at the bulk level. We have since, given the realization that we had with microglia in Alzheimer's disease, we have since started looking at single cell dissection of these epigenomic and these transcriptional changes. We've looked at many different traits, including many brain-related diseases, such as Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar, Down syndrome, autism, and many other disorders, as well as obesity and metabolism and cardiac disorders, etc., at single cell resolution across thousands of human postmortem samples, as well as living brain from epileptic surgery and living heart from open heart surgery. We've discovered many different subtypes, dozens of subtypes of neurons, both excitatory and inhibitory, many subtypes of astrocytes, more than a dozen different subtypes of microglia, many different subtypes of vasculature cells from the brain, as well as from many other tissues in the body. We've looked at multiple regions and more than 1,000 RNA and more than 500 single cell attack datasets. In the first study that we published 2 years ago in Nature in collaboration with Li Guizai, we basically studied 48 individuals across 84,000 cells, including both pre-symptomatic with high cognition and no pathological signatures, early Alzheimer's, and then late Alzheimer's with dramatically reduced cognition and many neurofibrillary tangle and amyloid A beta signatures. What we found is that it's not just the microglia that change. Every single major cell type shows dramatic alterations between disease and non-disease individuals. We also found that in female individuals, the cells from female individuals were dramatically enriched in the AD-associated subsets of every major cell type, suggesting that the transcriptional burden is much higher for female individuals. We find that early in the disease pathology and the disease progression, we see dramatic cell type specific changes, but later in the disease progression, we see much more shared changes, many of which are in fact associated with response to the damage that is happening to the cells. And we found dramatic differences between men and women, with 3,000 genes differentially expressed between men and women prior to Alzheimer's, but 6,000 genes differentially expressed after Alzheimer's, suggesting that there's dramatic sex-specific differences that we have to take into account as we develop therapeutic approaches against these disorders. We specifically found that men upregulated their myelination pathways in oligodendrocytes when women did not, leading us to the hypothesis that there would be more white matter loss in women. And that's exactly what we found for brains of female subjects. We have since expanded to 1.6 million brain cells across 7 different brain regions, each in 48 individuals, giving us access to marker genes for more than 30 excitatory subtypes and 23 inhibitory subtypes across prefrontal cortex, midtemporal cortex, angular gyrus, entorhinal cortex, thalamus, hippocampus, and mammillary body. We found that neurons vary dramatically across these different brain regions, especially excitatory neurons, but also inhibitory neurons. But dramatically, we found that glial cells also vary greatly. Here's astrocytes, where in the prefrontal cortex, they show very different signatures from mammillary body or from thalamus astrocytes. And most importantly, we're able to take all of these 1.6 million cells and for each of them make a predictive function that predicts whether the individual is Alzheimer's or control using the classification of the individuals that the cells came from and also the local pathology in every brain region that the cells came from. And using this predictive signature, we're able to classify every cell as more transcriptionally advanced in Alzheimer's or transcriptionally healthy, and paint a time course of all of these millions of cells across brain regions and across individuals to understand what are the genes that act early, intermediate, or late in the disease progression for Alzheimer's disease. Beyond the single cell data that breaks all spatial context for those cells, we found that the transcriptional profiles in the single cell data sets allowed us to capture sub-regions of the hippocampal formation, including CA1, 2, 3, 4, dental gyrus, subiculum, and then multiple regions and layers of the entorhinal cortex. These clustering was discovered purely de novo, and yet we found this beautiful concordance with pyramidal and granule cells and so on and so forth. And we found that CA1 is in fact the most predictive of Alzheimer's, whereas dentate gyrus appears to be the least predictive and most likely the least affected. We are also able to take spatial transcriptomic data sets, which I like to call mini bulk experiments, which are capturing multiple cells in every one of these little pucks, puck pixels, and then deconvolve it using this program that one of my postdocs developed previously, CDSeq. And we are able to distinguish changes that are happening between the proportion of different cells. For example, in the cerebellum of SCA1 mice, we see this dramatic loss of Purkinje cells that is simply not visible if you use instead the cell type marker of PP1R17, which appears to be normally distributed, but the identity of these cells appears to be lost and dramatically reduced. Prompted by all these findings, we basically developed a deep learning algorithm that allows us to now start predicting the spatial positioning of individual cells across neurons and oligodendrocytes and astrocytes and even microglia, that is in fact inferring the spatial positioning of all of these cells and their relationship to each other, enabling us to now start studying correlated patterns of change that are happening in the microglia and the glial cells that are surrounding individual neurons that are coordinatedly changing across these large data sets. Beyond Alzheimer's, we've been looking at schizophrenia and able to predict for more than 100 different genetic regions that are loci associated with schizophrenia. What are the candidate target genes in these loci that are differentially expressed in schizophrenia individuals? And what is the directionality of change? And what is the specific cell type where they're changing? And what we found is that indeed, we are able to annotate the vast majority of the genetic loci with genes that are very often supported also by three-dimensional linking information from chromatin conformation capture. And we're able to predict which individuals show schizophrenia based on their global transcriptional signatures. Here, for one control individual, we predicted that they should be schizophrenic based on their transcriptional signatures. And indeed, their son was diagnosed with schizophrenia, even though they were not diagnosed. At the other end of the spectrum, we have 4 individuals that show transcriptionally normal signatures, but are instead enriched for this specific SZTR cell state, which appears to be there in individuals that are diagnosed with schizophrenia, but are in fact showing otherwise normal signatures. And there's 2 interpretations for this, either an alternate path to schizophrenia based on SZTR, or perhaps a transcriptionally protective state, which leads to resilience for these individuals at the transcriptional level, even though they're diagnosed. We're able to use this information to start predicting upstream regulators and when they're acting in development. And we're finding that they have both synaptic functions and neurodevelopmental functions. We're able to carry this out for psychosis in the context of AD. And what we're finding, in contrast to schizophrenia, is that psychosis in Alzheimer's is associated with loss of excitatory and inhibitory neuron function rather than overactivation, as we see in schizophrenia. And there's many pathways associated with ribosome biogenesis, neuronal projection, autophagy, et cetera, that are dramatically shut down in the context of psychosis in AD, which appears to predominantly affect women, whereas psychosis in schizophrenia appears to predominantly affect men. We've done this for ALS and FTD, and we find very common pathways and very similar signatures. For Huntington's disease, we're finding cell types that lose their identity between direct and indirect pathways, spinal projection neurons. We're able to look within cell-sorted cells, in silico cell-sorted across hundreds of thousands of microglial cells to distinguish synaptic microglia that appear to be associated with schizophrenia, both for GWAS in the single-cell attack data and for differentially expressed genes in the single-cell RNA data, and inflammatory microglia that appear to be instead associated with Alzheimer's in both data types. We just recently posted a paper dissecting the vasculature of the human brain at single-cell resolution and discovering multiple zonation signals in endothelial cells, in neural cells, including pericytes and smooth muscle cells, and how these are changing in the context of Huntington's disease. And we're able to link, by coordinately profiling single-cell attack data and single-cell RNA data, non-coding variants associated with schizophrenia with candidate target genes based on their correlated activities. And you can see here the coordinated changes are happening in the context of control versus disease individuals in Alzheimer's, where you can see both transcriptional changes and coordinated changes in DNA accessibility in these diagonally pointing arrows, both up and down, where 75% of these are in fact consistent in their changes. We're able to use this to now start localizing genetic variants in Alzheimer's into microglial cell states and in schizophrenia into excitatory and inhibitory neurons, and start fine mapping these regions to individual motifs and individual regulators enriched in each of those. And then lastly, we're able to look at how mutational signatures of somatic mosaicism are accumulating within these single-cell RNA-seq data from SMART-seq, that's looking at the whole transcriptome, and how these are affecting specific cell types, specific both male or female individuals, differences in amyloid, and also different pathways in each of these cell types. In a paper that we just posted in bioRxiv a few weeks ago, we've basically dissected the QTLs at single-cell resolution through global expansion and deconvolution of 3,000 brain samples. And again, pointing to microglia as the causal cell type. And we've also looked at different signatures of disease and how they're associated with different regions, expanding into the medical record. In a paper that we recently published, we're able to combine multiple of these phenotypes and then start predicting drivers that are genetic and associated with gene expression changes in specific cell types for specific groups of individuals grouped based on their electronic health records. In a paper that we just presented at Cold Spring Harbor last week, we basically have been looking at multiple tissues and their response to both diet and exercise in both human and mouse. And we're finding these coordinated changes across multiple tissues, these thermogenic tissues that I mentioned earlier, and again, implicating early adipocyte differentiation and muscle and their coordinated changes through adipocyte stem cells and T cells that are shifted in opposite ways and that are controlling multiple nearby cells, both within the same tissue and across tissues in exercise and obesity in opposite ways. And then lastly, we've basically developed many methods for dissecting these regions systematically using modular and programmable CRISPR-Cas9 constructs to edit individual variants, to knock out promoters or target genes, and to repress or activate different enhancers in iPS cells and then differentiate it into these different cell types. We've also developed methods for massively parallelly testing millions of accessible regions dissected directly from attack experiments, inserted in self-transcribing constructs, enabling us to now start fine mapping the enhancer activity into individual evolutionarily conserved and motif-containing sites that are predicted to be drivers. And then we're using massively powered reporter assays and PRA technology in the context of mouse brain with Andreas Fenning, a previous postdoc in the lab who's now at TMU, to start understanding the spatial context in which these genetic variants are acting. So I'll acknowledge an amazing team of individuals who are very interdisciplinary, both experimental and computational, a beautiful collaboration with Li Hui Tsai, with whom many of these projects are carried out. And many of the postdocs have now started off their own labs. So we're looking for postdocs. So please let us know if you're looking for a position and send us your best and brightest. So that's where I'll stop, and I'll be delighted to take any questions if there's time.
Excellent. Thank you, Manolis. Yeah, I think so. We have a couple of questions. That was a whirlwind tour. The last 5 minutes was an amazing amount of data you went through. Anyway, so there are 2 questions online that people posted, and if anybody has questions, they should put them into the chat on the web interface. The first one is from Rachel Massey. So the question is, what population of persons or database was used for the GWAS, and is the data applicable to non-Europeans? So I'm not exactly sure which GWAS that referred to, but
we've basically taken the entire GWAS catalog across all ethnicities, including Asian Americans, Asian, African ancestry, and European ancestry samples. Unfortunately, the vast majority of these, about 80%, are European ancestry GWASs. And that's something that the whole field has recognized is a dramatic shortcoming, both because many of these individuals from lower socioeconomic status in the US, for example, are of African or Latin American descent, thereby exacerbating both their lack of privilege at the phenotypic level, but also their lack of representation in the genetic databases. And number 2, African ancestry individuals capture so much more variation per capita than European ancestry individuals that are a tiny little subset that went out of Africa. So we are basically shooting ourselves in the foot by not systematically including multi-ethnic, multi-ancestry samples that truly capture the whole human variation. So it's a shortcoming of the field. We acknowledge it, and we are working hard to remedy it.
Excellent, thank you. So the other question online was from Mike O'Neill. It says, have you been able to identify any germline epigenome signatures associated with transgenerational epigenetic disease or physiological states?
Wow. So I'm cautious to use the word epigenomics because I don't want to make any connotations of inheritance that the field of epigenetics, unfortunately, is fighting with. There are many mechanisms through which epigenetic inheritance can happen, where on top of the genetic information, there can be additional inheritance. At the epigenomics side itself, there's 2 rounds of reprogramming, both in gamete formation and after zygote formation, to wipe out any epigenomic memory. However, small RNAs and large RNAs can still exert effects on the new generation based on epigenetic memory from previous generations. There's an incredible diversity of activities trying to dissect this currently in our field. It's a major area of investigation as we're trying to understand the impact of environment, not just in our current lifetime, but also for our children based on the exposures that we are exposed to. This is not something that is visible in the current data that we have profiled, but it's something that I think the whole field will be heading towards. And stay tuned, there will be more information on it.
Excellent. So I have one question from me about the coronavirus stuff you talked about. So you described this new ORF3C, I think, which is newly described. So was that an ORF that's only present in a subset of coronaviruses, or it was just a newly recognized ORF?
It's an excellent question. So so if you look at the transcriptome of the SARS-CoV-2 virus, and more generally, the coronaviruses in that family, you see there's a separate transcript for, of course, ORF1ab, and the host protein only translates one protein from every transcript. It doesn't like to sort of start in the middle. ORF3a is the transcript from which this happens, but you can see here that ORF3c is just a little downstream of that. And in fact, the stop codon context, the start codon context of ORF3c is very, very strong as well. And what you can see here is that across all of the 44 cervical viruses, these are the SARS-related beta coronaviruses, portmanteau for that is cervical viruses, which we analyze. You see this beautiful conservation across all of them. So this indicates that this is not just an ORF that was recently born in SARS-CoV-2. This is an ORF that has been around for a long time. It was there for SARS 2003, and it was simply not previously recognized because, frankly, nobody looked at this through the lens of our phylo-CSX program that we developed many, many years ago, because I guess we were not stuck all at home wondering how we can help with the current pandemic. But this is not a new ORF, and moreover, our methodologies are specifically tuned for discovering things that are conserved across the related genomes, and therefore, if we found it, it's probably that it's conserved, but we see that there are no stop codons in any of these other strains. You can see here the stop codon alternation between TAG and TGA and TAA, which you can see here. That indicates, again, the pressure to preserve the stop codon, even though the specific stop codon used can be changed. And you can see here the perfectly conserved ATG start codon across all of them. So yes, it is a conserved, deeply conserved ORF.
Okay, excellent. Well, thank you. So I think, unfortunately, I think that's all the time we have for questions. But I want to thank you again for your wonderful talk. As I said, a whirlwind tour as usual. And at this point, I'll turn it over to Rachel O'Neill, who will wrap things up and introduce the poster session. So thanks, Manolis.
Thank you. And feel free to email me, guys, if you're looking for postdocs. We're very, very hard looking for both computational and experimental people. So see you at the bar later on, right, Brent?
Excellent, yes.
All right, thank you, everyone. And thank you, Manolis. That was exciting. And so now I'd like to direct everyone to the